So when we talk about how to organize our code, we often will talk about R projects. So basically, an R project is in RStudio. When you look at this tab on the upper right, that's what's telling you what project you're working in. They're a really useful way to organize your code. And in fact, if you're, for example, writing an R package, they're essential for you to keep all of the code that belongs in your package together. So just as kind of an example of this, I'm going to show you how you might make a new project. So I'll make a new project here. So let's say it's a new directory. They actually have preset project types that you can pick from, which is really nice. So I'll just pick a basic R project since I don't have any need for anything beyond that. And so let's say that I make this project in, I'll make it in our course folder. So I'll make it in here and I'll call it example R project. And I can just go ahead and create that here. So right now, these are the files that are contained in my R project. There's not a whole lot here, but there is my .rproj file and this folder that contains information about the project. You don't really have to know how these files work or the folders work, and the reason is because most of this will be essentially automated so that you don't have to customize it yourself. So the first thing I can do is I can make some code. So I'll call this my script, and it'll just print hello world. Okay, and we can see that when I saved that script, it automatically got saved in the project folder. Now, we might want to also have some data in our folder. So maybe I'll make another script here called get data. And this will load the iris dataset into the global environment here. So I'm sure you've had a chance now to work with iris because of the hands-on activity from last time, but if you recall, it's got information about flowers and of these different species here. So maybe I'll save this. I'll save this as a CSV file. And we can already see that because I'm in a project, I could just save this like this without having to specify any sort of file path. And right. And you can see it ends up right in my project folder. So why is this cool? Well, this is cool for a couple reasons, but the main one is because, let me, don't save. The main reason is because when I now go to that folder, my example R project, I can go to this folder and I can double click on the rproj file, and now it will open up my project exactly how it was when I closed it. So all of the same scripts are still open, even the iris dataset is still here, and all of my data and files are right here ready to go. What's really cool about this is that I could actually take this folder and I could move it. I can move it to my desktop. So I'll move it here to my desktop. And now it's in a totally different place in my file system. But just like before, if I open up that rproj file, and it just takes a second, now everything opens up just like it was before. And you can use it exactly how you used it before. I don't know why this is taking a second to load, but oh. There it is. So exactly how it was before, even though I've totally changed where that folder was in my file system. So why is this powerful? Well, this is powerful because you could take your R project folder and you could email it to someone. You could put it on a flash drive. You could do whatever you wanted to with it, and all of your code would still run just the same as it did before. And the reason is, of course, because pretty much everything is exactly how it was. So if I wanted to load this CSV file, I don't have to know the actual path of that CSV file on my computer. All I have to know is that this CSV file is in my project folder. And as long as I'm in my project, then it's right here, ready to go. So Catherine asked, are there packages you loaded for this project already there as well? So if in my script, I also had, I needed to, for example, load, ggpubr. If I needed to do this in my script, then it would be, this line would stay in my script. But as far as I know, this wouldn't keep this package loaded in my environment. Although I suspect there's probably a way to do that. Simon says, can you create subfolders in your projects? And will they move along with yeah, they will. So everything, as long as so it's kind of like your root directory now becomes wherever this rproj file is. So right now I'm in the root of my project. And so I could make a new folder. I could call this my folder. And then I could save some data in it. So I'm going to write some data into it. I'll write mtcars. And I'll put it in my folder. So now we can see that I've written mtcars into my folder here. Sure. And so again, I'll just show you that this is still true. I can take this and now I'm going to put it in my, I don't know, I'll put it in OneDrive, I guess. I'll put it in OneDrive. Oh yeah, it doesn't like that if you have it open in RStudio. Don't save. All right. Okay, so I'm going to take this. Now I'm going to put it into OneDrive. And you'll see here that when I open up this rproj file, and I saw this just takes a sec. It's still got the folder and the data set are right there, just like they were before. So if I had any code that relied on this data, that code would work no matter where this folder was on my file system. Even if I emailed it to someone, if I zipped this, if I take this and I zip it in like a zip drive and I email it to someone, it will still work exactly the same on their computer as well. So that's kind of the power of the projects and why we should, or at least I personally think we should try to use them as much as possible. Not everyone does, but I like them. And you can close a project very easily. So you just come over here and you click Close Project. So this brings us now to the week 5 data. So week 5 is actually a folder containing an rproj file. So what you're going to do is you're going to take week 5 and you're actually going to download the whole thing now. And so it downloads as a zip drive. Should just take a second. All right. I don't genuinely know on Mac if it works the same way, but on Windows you can just extract this into your downloads folder if you want, or wherever you want to extract it to. All right, and now I've got my week 5, including the week 5 notes, ready to go. So hopefully you all did this like this. The reason we wanted to set it up this way for this week is because you need to read in the data from this, this file here. And so you don't have to now manage that file path. It should work exactly like it's written here because it's in the same folder that you're in. And just to prove that here, I'm going to load the tidyverse, and then I'm going to actually read it in. And in fact, that did work. So, okay. I'm going to just really briefly go through the lecture. And oh, I guess I already had it open. That went to the wrong screen. All right, I don't know why this is frozen. I'm sorry. 1 second. Because this PowerPoint is 20 gigabytes in size, or 20 megabytes in size. It's kind of odd, but okay. So I'm not Victoria, but I'm going to do my best to give her presentation here. So you shouldn't have to follow along with the notes in RStudio yet. It's just going to be a really brief set of slides, and then we're going to jump right into the R code. So let's talk about when you should use R and when you shouldn't, because it's not always the right answer for every data analysis problem. So this is just a nice little comic about that. So many of you are probably familiar with using Excel. You can very quickly do some basic analyses and statistics in Excel, and you can even create basic graphs. But some cases require more. Some cases require you to have R, where you can actually deal with large and sometimes messy data. It's also interactive, and you can make more complex graphics. You could also make a much nicer data analysis report doing it this way. So understanding which packages to pick is also important. Not always, oh, and I should mention, we're going to be using this for qPCR analysis today. So the first thing is to understand what are the objectives of your analysis. And so in our case, we're going to be trying to analyze qPCR data. The next step is to actually sketch a workflow to understand what are the discrete steps of your analysis. Then pick the correct packages, and finally create your script and debug it. So I think she actually wrote this herself. Yeah. So this is her sketching the analysis workflow for the data we're going to be talking about today. So basically, she did an experiment here where she was looking at U2OS cells. So for those of you who are familiar with cancer biology, U2OS is a kind of osteosarcoma. And she was looking at, I think it's the gene is OGG1, which is a gene that I think they believed was relevant to the progression of these cancer cells. And then she was using siRNAs to knock down that gene and measuring the expression using RT-qPCR. So there were several different siRNAs and several different primers for the gene, along with GAPDH, which is their housekeeping gene. So I suspect several of you have probably done RT-qPCR before. You'll probably recognize this sort of design. It's pretty standard and straightforward for analyzing expression of RNA. But for those of you who aren't familiar, do feel free to ask if you have any questions. Do feel free to ask for more clarification, and I'd be happy to stop and give a little bit more clarification on this. So basically, once you do your analysis, you get, and I'm actually just going to show you because I'm not sure this is actually in this PowerPoint, but you get a file kind of like this. So I'm going to open this with Excel so you can see what's going on here. So this is kind of like what the machine will give you back. So it'll include information from the PCR machine, such as your plate ID and your file name and your run date, run time, and then it's going to include a lot of extraneous information about the wells of your plate and the type of reporter you used, and so on and so forth. Ultimately, what you probably care about more than anything else really here is this CT value. And we're going to talk about that in a second, how you get that and how you analyze it with R. So there was just more information here. So this is your GAP-DH primer, and then this is your OGG1 primer, OGG1 primer, and so on and so forth with the other primers. You also have information here in the sample name column about what was the cell line and then what was the type of siRNA. So this is 3 replicates using a scramble siRNA. This is one set of replicates using siRNA 1, 2, 3, 4, and 5, and so on. Does anybody have any questions about what we're looking at here, or kind of the outline of this experiment? Okay. So the question is, how do we actually analyze this in R? And then how do we do things like the delta CT and the delta delta CT calculation, and then actually visualize that data? So she sketched here a workflow to do that. So the first step was to tidy the data, then to transform it, and that's where you're really getting your delta CT and your delta delta CT calculations. Then to visualize the data with a bar graph. Oh, right. So this is delta CT is up here. Then she wanted to do delta delta CT down here, visualizing the delta CT, and then coming back and visualizing the delta delta CT. So the next step is to actually pick the correct packages to do this with. And this includes things such as readr and tidyr and dplyr. So basically here, this is showing the workflow with the packages overlaid on top. And so let's actually do it now. So how do you analyze this data? Well, the first step was to actually read in the data using readr. So to get started, you have to first load your tidyverse packages. And then the first step is to actually read in the data. So she used here this read_delim command. If you have any confusion about what that is or what it's doing, you can always use the question mark read_delim command, and this will actually tell you information about it. So basically, it's saying read a delimited file, including CSV and TSV, into a tibble. In this case, our data is a TSV. And we know that because if we open our file, and I'll just show you here, if we open our file with a text editor, like a plain text editor, WordPad's a good example of this. That's right, I have it open in Excel. Funny thing about Excel is it doesn't like you to open stuff if you already have it open in Excel. So we open this and we can see that all of our columns are here and everything is separated by a tab. So that's what makes this tab delimited or tab separated, as opposed to comma separated, where instead of a tab, it would be a comma. All right, so that's what makes it a TSV file, or in this case, TXT with a tab separation. So if we rename this to TSV, that would mean the same thing. It just means that it's tab separated values. All right. Basically, some of the other, and I'll actually add this here so it's a little bit more clear what this is doing. Delimiter. So basically, when we do read delim, we tell it what file we want. So this is the file here. We tell it what the delimiter is. So delimiter meaning what is separating the values in your text file. In this case, it's a tab, and this is what this is how you say tab in R and in other programming languages. It's \T. That just means tab. If it was comma separated, we would put a comma here. But since it's tab separated, we put a tab. Escape double. So I don't actually know what that means. So I'm going to look over here, and I can see escape double means does the file escape quotes by doubling them. So I don't actually know if this is necessary here, but it might be. So this is one of the options you can set. Trim WS means trim white space. That just means that if you have extra white space in your document, it'll trim that away. I don't actually think that one's necessary here either, probably. And then skip means how many lines should you skip before actually reading in the data. So why would we want to skip lines? Well, there's a pretty good reason, actually. So if we go and look at our text file again, and we'll open it with Excel this time, we can see that the first 9 or so lines are not really relevant to our data. We don't care about these lines of the file. So we're all right with skipping them when we read in the data. So that's why we skip 9. So when we do this, we get some warning, missing column names filled in. So let's see what it actually did there. We can see here that even though we skipped 9 lines, we probably should have skipped 10 because we can see that our column names are for some reason on our first line of our data. So let's skip 10 instead. And now we didn't get a warning, and we can see here that now our headers, our column names are in the correct spot. Does anybody have any questions about how I'm doing this read command and what the options are telling us? Yep. Yeah, you can use the, well, I think you can. So if we look at read Excel, it looks like that's from, oh, I see. There it is. So are you talking about like read XLS, or are you talking about a different function? Okay. So read XLS, you can see here when you're looking at the arguments that are available. So let's actually put this in here and see what the help page tells us, because I don't know offhand the answer to your question. So you've got, yeah, so read Excel and read XLS and read XLSX all have a skip argument. So you can use skip with them as well. Okay. So now we've got our data, and let's take a look at it here. We can see here that if we're kind of expanding it out, there's a lot of NA columns. We probably don't care about those. And there's a lot of space where the data has just kind of disappeared. And of course, that's because if you'll recall in our Excel view, there were areas where this is kind of like, it's almost like a decorative part of the table. It's really not meant to be real data, but just kind of to separate your gap DH from your OGG1, and then separate OGG1 from OGG1-2, and so on and so forth. So you need to get rid of that for this to be usable in R. There's a few other things we'd like to clean as well. So we'll jump down here to step 3, which is to tidy the data. Now, we're going to go through this a little bit slowly just so you can kind of see where we're going with each of these steps. So for the first step, we want to select only the data that we want to analyze. So in this case, those are the columns sample name and detector name and CT. because as we can see here, sample name contains our sample names. Detector contains the primer, so GapDH versus OGG11. And then CT is our CT value. That's ultimately what we're going to care about as we analyze the data. So we're going to select only those columns. And we're not just going to select them, we're actually going to reassign them to new column names. So sample name will become sample, detector name will become primer, and CT will stay the same. So if we just take this part by itself, let's see what that does. So we can see here that what it's done is it's taken our sample name column, it's renamed it as sample, it's taken our detector name column, renamed it as primer, and then it's kept the CT column. And we've only kept these 3 columns. We got rid of all the other columns. All right, so that's what this first part's going to do. But then we're actually doing another pipe. We're actually saying we also want to get rid of the empty cells and rows. And we use drop NA to do that. So that's going to drop all the rows where you have NA values. So originally our data is 111 rows by 3 columns. Let's see here now what happens. So now it's only 87 rows. So we dropped all those rows where there were a bunch of NA values that we didn't care about. All right, so now we want to keep the rows that do not have a detector name in the primer column. So basically, if you look here, we can see that when we're coming down here, we drop these rows, but there's actually another row of column names when you get down here. And you don't care about this. This isn't data. This is just another set of column names. And then when you get down here, there's another set of column names. So what we're saying with our filter statement is we want to get rid of the rows where primer yeah, so we're getting rid of the rows where primer does not equal detector name. Do not have detector name in the oh, I see. Yeah, we're getting rid of the columns where I'm not sure what she's saying here. I don't know why we would do that. Yeah, so where it does not equal detector name. And I guess that worked the way she had it, but that's not particularly intuitive. So we want the rows where primer name does not equal detector name. So basically that means we're getting rid of these rows. We're only keeping the rows that don't have this. And then we're getting rid of the rows where CT does not equal. That's kind of redundant. We don't actually need to do that. All right. So now we're going to get rid of the rows that have the header. And I'll actually assign this to a new variable so you can see this. So you can see we've now only kept the rows that have real data in them. But there's still a problem, right? There's these columns where you have undetermined as your CT value. You don't really care about that. So what we're going to do is we're going to do another filter, and we're going to say only keep the rows where CT does not equal undetermined. So again, we're going to do this, and I'll assign it to a variable so you can see it. So now we've gotten rid of all the rows where undetermined was the CT value. But we actually have another problem, and you'll see it here. When we look at our new filtered data, we can see that sample is a character, primer is a character, that's all correct, but CT is also a character. That's actually wrong. We'd like CT to be a numeric value because we're going to want to be able to do mathematical operations upon it. And you can't do mathematical operations on a character. So we'll come over here and we'll do our very last dplyr line, which is mutate at. So we're basically saying mutate at CV, and we're saying, basically what we're saying here is take the column CT and apply a function to it. And the function we're applying is as.numeric. So that's going to take anything in R and turn it into a numeric. So we're basically saying take this column and across the entire column apply this function, turning every element of the column into a numeric. All right. So now if we glimpse, and glimpse is just a way to summarize the tibble and kind of look at it in that summary form, we can see that we've got now 3 columns, 72 rows. Sample is a character, primer is a character, and CT is a double, which is just another way of saying a numeric. All right, that was a lot to go through. Does anybody have any questions before we jump into hands-on activity number 1? Okay, so hands-on activity number 1 takes just a tiny bit of explanation. So basically, I found some qPCR data online, and if you, I'm just going to show you. I'm going to show you it here. So this is where I got it from, and you can actually see here if you go to their GitHub repository, this is where that data lives. So basically, just like I showed with our qPCR data, this data also has that sort of plate information up here, and it's got kind of like we showed before, it's got your detector and sample name and all of that same sort of information. And it also includes areas where you don't have any real data that you need, and you need to be able to remove those rows. So let's take a look at this in R now. Something else that you have to keep in mind with this data that makes it a little bit different from the last time is that they don't automatically have the primers matched up with the rows of the data frame. And they also don't automatically have the treatment type matched up either. So what we do have is we do have wells, and that tells us so if you're looking at a 96 well plate, this would be well A1, A2, and so on and so forth. And we know what treatment corresponds to each row and each column. So that's basically what we have here. We know the treatments that correspond to the rows, or sorry, the primers that correspond to the rows. And we can see this here. So row A has a housekeeping gene, and row E has the test primer. And we know how the columns relate to the treatments. And so I've already given you this information. We basically know column 1 is treated with RNAi1, and so on and so forth. So when we load in the data, we can see it here. It contains all of that information, and it also contains the wells. So the first thing that I do, and I'm just going to really briefly walk through how I did this. The first thing I do is I want to take the wells, and I want to get the row from this. So the row, of course, is the A, B, C, D, E, F. And then the column of the 96 well plate is the 1, 2, 3, 4, 5, 6, 7, and so on and so forth. So to get the row and assign it to a new column, I use the substring command. So what does the substring command do? Well, it's going to, I'll just show it here. It's going to take, so for example, if I had a vector that was AAA, BBB, ABC, if that was my character vector, if I did substring on the character vector, I'll say start at 1 and stop at 2. So what is this going to do? Well, it's going to take the first 2 elements. It's going to take all the elements, or sorry, all of the characters in the string from 1 to 2. So AA, BB, and AB. Does anybody have any questions on how the substring command is working? Okay. So basically what I'm doing here is I'm taking the substring. How would you deal with double-digit columns? That's a great question. Well, I probably wouldn't. So I wouldn't use this command if I had that kind of situation. I would actually use regular expressions if I had that kind of situation. But unfortunately, that's a little bit too complex for this lecture. I hope, though, in the future we'll get to do like a short workshop on how to do regular expressions in R, because honestly it's a very valuable skill. But without regular expressions, this is the way we can do this. So we know that in the well column, that the first character in the string in that column is the row, and the second character in the string is the column name, or is the column of the plate. Hopefully that makes sense. So if I actually do this here, and I look, and I'll just assign this to a new data object so you can see it more easily. So if I run these first two commands here, well, I can see if I've got if I've got well A1, then that's row A, column 1. So you think about 96 well plate, A1 in your plate is going to be row A, so that's actually the first row, and then the first column. And if I go down here to B8, I can see that's row B, column 8. So I know I said this was going to be this is going to be an interactive hands-on example, and it will be in a second, but I feel this part deserves a little bit more explanation. So why did I do this? Well, because I need to now merge my qPCR data with my primer key and my treatment key, because I need to be able to match up these treatments to the columns in my qPCR data. So basically, where I have column 1 here, I need to annotate this row so that it's known that this column corresponds to this treatment. And where I have primer, or where I have row A, I need to know that that corresponds to the housekeeping gene. So in this case, I've got row A, and I now need to annotate this so that it's corresponding to the housekeeping gene label. So basically, what I need to do is I need to take this qPCR data and merge it with these two data frames here. And the way I'm going to do that is I'm going to use a join. If you've ever heard of a join in like SQL or in some other context, it's basically exactly what's going on here. And I'm just going to show you, if you go to the lecture notes, I'm sorry, not the lecture notes, the resources, you can see here that I have provided a dplyr cheat sheet. And I'm just going to download this and open it because it's actually got a really great visual representation of how a join works in dplyr. I'm actually going to choose a different one, because I think that there's probably an even better one right here. Yeah, I'm going to include this in the resources folder in just a second. But if you Google dplyr cheat sheet, it's literally the first thing that shows up. So you can also find it yourself doing that. Basically, this is how this works. If you've got two data sets, let's call them A and B. And let's say that they've got one column in common. So both A and B have this X1 column in common, but they have another column that's different. So what I want is to add these data sets together. Basically, I want a new data set, for example here, that includes X1, X2, and X3, where X2 and X3 have essentially been joined together by x1. So because both of these data sets share x1, you're able to match x3 to x2 by using this x1 column. So, A corresponds to 1, A also corresponds to true, therefore A corresponds to 1 and true. And that's called a join. Does anybody have any questions on what a join is or how this is working? So Aaron asked a really good question, which is why do you bother using like a left join versus a right join? I honestly don't know. I don't use these. I typically use only inner joins or full joins. So an inner join is where, if for example you have this, in this case it's NA, if you're joining on this X1 column, I've got A, B, and C here, but I have A, B, and D here. C is not in this data set, and D is not in this data set. Therefore, if you do an inner join, it's only going to keep A and B because those were the rows that were in both data sets. I typically only do that, but sometimes it's beneficial to keep, for example, even if C isn't in this data set, it's beneficial to keep it in this data set. So that's why in this particular case, a left join will give you A, B, and C. That's because the left side, they kept all of the values from the left side. So A, B, and C were kept, but D, which was on the right side, was dropped. That's okay. I'm gonna get to that in a second. So if instead I did a right join, it'll keep everything in this data set. So it keeps A, B, and D, but it drops C because C wasn't in the right side. So the question was, why don't you just do a left join and just reverse the order? Yeah, that you could do that. That would be the same thing as doing a right join. Hopefully that answers your question there. Does it do one-to-many relationships? Yeah, it does. So you can easily do one-to-many relationships. So for example, if I had, and actually this is going to be an example of that right here, because we can see here that we're about to do a left join on qPCR data and primer key. Now primer key is only 8 rows long, and qPCR data is 96 rows long. So let's look at these. qPCR data has a lot of wells. And so primer key has only 8 rows because it only accounts for 8 rows in the well plate. And so, for example, where we have A1 here, A2, A3, that would just be A in terms of plate rows. So when we merge this with the primer key, A is going to be a one-to-many join. So basically, this row from primer key is going to be joined to all of the rows in qPCR data that have an A in their well column. Does that make sense, Simon? I guess, does that answer your question? Okay. And likewise, with the treatment key, we've only got columns 1 through 12 here. We're going to join the treatment key to qPCR data. So where you've got a 1 here, it's going to be joined to this row. So where you've got a 1 here, it's going to be joined to this row as well. So it's kind of a one-to-many, as we kind of talked about already. It's not crucial that you understand this at this point. It's very beneficial to understand how to use these joins as you go forward in the future. But all that being said, the end result is that you've now got, for all of your rows and columns, you've now got your primer and your treatment annotated. So we can see here that this works exactly as we intended. And it's not necessary that you understand how to do this, but it's just kind of cool that you can, using ggplot, you can actually visualize your plate. So for example, these are the columns of our plate, and these are the rows of our plate. So imagine yourself looking at a 96 well plate. This is kind of what it would look like. And we can see here, this is our different replicates across here for our RNAi1 treatment in our housekeeping gene. So red is housekeeping, blue is our test. Does anybody have any questions about how we got to this point or kind of what this is representing? Okay. So I think that this sort of visualization is really useful because it makes it easy to visualize this as an actual, PCR plate. So we can also add a title to this. That's the next line down. And then we can also add customized X and Y axis labels. And that's the next line after that. And then finally, we can mess with the theme. So this is a way of adding the black and white theme to this. So I just add a plus and then theme underscore BW to do this. So what if we wanted to color by treatment instead of by primers? Well, we could just change the fill from primers to treatment. So hopefully that makes sense how we're doing this. All right. So for 15 minutes, please go ahead and attempt the questions here. Actually, I'll give you 10 minutes since it's really just fill in the blank. And feel free to post in the chat if you have any questions, and we'll meet back up at 6:01. Okay. Does anybody have any questions or concerns about that? Okay, well, I'm going to go ahead and go through the answers then. So, can anyone tell me what is the verb that should go here? I'll zoom in a little bit. Should go here. Okay, great. It is the select verb. And I think I gave you that, guys, this part. Yep. Treatment is... All right, so when we run this, we get our 4 columns just like we intended, with the correct names associated. All right, so we can just kind of copy and paste as we go along. Copy and paste is a great skill to have as a programmer. Can anyone tell me what the verb we use here is to remove the rows with NA values? Yep. Yeah, Kevin got it. Drop NA. Yep, Aola got it. Simon got it. Okay, perfect. So you can see here when we look at our data, there's like a row here with an NA value, right? We don't want that. So we're going to drop these NA rows. Now we don't have any more NA values. This is kind of squished. I'm going to zoom out a little bit. Okay, so the next step is to remove all the rows with undetermined as the CT value. All right, who can tell me what this verb needs to be? Okay, great. It is indeed the filter verb. So when we run this, we can see that all the rows that had undetermined have now been dropped. And we can see this because there are now, instead of 72 rows as there were before, there's now 70 rows. All right, finally, or I shouldn't say finally, it's third to last. We need to remove the rows that have NTC as the sample. So who can put in the chat the command to do that? It is a filter. Can anyone tell me like yeah, so like how Simon was saying, I guess I should be more clear. I was asking for the actual filter statement. So yeah, it is filter sample does not equal NTC. And basically what we're doing now is we're getting rid of the rows where CT is undetermined, and we're getting rid of the rows where sample is NTC. And so now we've only got 44 rows left. And then finally, we want to force the CT column to be a numeric value. Can anyone tell me what the verb to do that is? Yep, exactly. It's mutate at. And just as kind of a challenge, can anyone tell me how mutate at is different from mutate? Exactly. Yeah, so whereas mutate is going to make like a new column, mutate at is going to apply something to the column in place, essentially. So we're basically saying, yeah, So we're basically saying that with mutate at, we're going to take the CT column and we're going to apply the function as numeric to it. All right, so finally I said to return this as a variable, tidyPCR. And now we've got our tidyPCR ready to go. Okay. All right, and then glimpse. So glimpse is going to show us now each of our columns. It's going to tell us what type they are and going to give us the first few elements of each along with the number of rows. Okay, did anybody have any issues with that or any questions about what we did there? All right, well, if not, then I'm going to move on to step 4, which is our data transformation. Basically, what we're going to do now is we're going to do the delta CT calculation. And for those of you who are unfamiliar with delta CT, I can just kind of briefly explain. So delta CT is going to be the difference between the CT value for your primer, your test primer, and your housekeeping primer or your control primer. So in our data frame, I'll just view it here, view tidy PCR. We can see here that we've got housekeeping primers and they've got CT values associated with them. And we have this for each of our treatment conditions, right? But we also have our treatment primer. And we have a treatment primer for each of our treatment conditions, or sorry, a test primer for each of our treatment conditions and a CT value. And what we need to do to calculate delta CT is we need to find the difference between this CT value for this treatment in the test primer and the CT value for that same treatment in the housekeeping primer. And so this is basically telling us the difference between our level of expression for our gene of interest and a housekeeping gene, such as GAPDH. It's a pretty straightforward calculation. It's just subtraction. You're just subtracting the two CTs. But what we typically want to do first is we want to find the average CT for the housekeeping gene within each sample group. So if we look at tidyres, this is now our data, our U2OS study. So if we're looking at tidyres, basically we can see here that you've got samples, you've got your SI control, your SI1, and so on and so forth. And you've got GAPDH, which is our housekeeping gene. And then you've got OGG11, OGG12, and so on and so forth. And what we need to do is for all of our U2OS replicates, these three here, for example, we need to calculate the average CT for the housekeeping gene. So basically we're averaging these three, and then we're averaging these three, and then we're averaging these three. And then we're going to do our delta CT by subtracting, for example, this value here by the average of these three values. So the first step is to actually get the average CT value for your housekeeping gene within each sample. And to do that, first we're going to make sure we're only getting the data where we're looking at the housekeeping gene. So we only want GAPDH. So this is what that would look like. Then you want to group by sample and then summarize it, because what you want is within each sample, you want the average CT value. So when we do this, we can see we now have the average CT value for U2OS SI1, SI2, all the way through 5, and then our scramble control. And this is really building off of the lecture from last week. Hopefully this is starting to make some pretty good intuitive sense. But definitely review the tidyverse lecture from last week if you're still having a hard time understanding how this group by summarize pattern is working. So the next thing we need to do is we need to now get the data that doesn't have GapDH, right? So we're basically saying here, I want all of the non-GapDH primers. And I want to do a left join with my HK gene, which is what I just created here, that contains the GapDH primers and the average for each. And now I want to do a left join so that for each sample, I know what the average CT for the housekeeping gene was. And then I can mutate to get the delta CT by subtracting the CT for each sample for the gene of interest by the CT for the housekeeping gene, the average CT for the housekeeping gene. So let's actually see what this did. So the first step was to get my average housekeeping gene CT, and so this is why all three of these have the same value, because that's the average value for my SI scramble sample. And I've also got the CT for every sample and every replicate. And then I simply subtracted these two columns from each other to get my delta CT. So this is just this minus this, gives you delta CT. Does anybody have any questions about how I did that, or any questions about how I got this data set like this? Okay. So then one of the things we can do is we can get the average CT within each sample and each primer. So basically that would be getting the average of this, the average delta CT, the average of this, the average of this, the average of this, and so on and so forth. And then also getting the standard deviation. So that this is getting your average. So take a look at this. This is now giving you your average. So you've collapsed your triplicates into one value, one delta CT value. And then for your standard deviation, which we're going to want to actually include on our plot, you can do a right join to get the average delta CT and delta CT together, and then do your group by, and then do your standard deviation. So this part, you don't actually need to do this for this to work. This can be done after the calculation. Whoops. Just so I hope this is a little bit simpler. To actually get your standard deviation, this is the same thing you were just doing to get your mean, except instead of the mean function, you're using the SD function, which stands for standard deviation. And then you're adding this back with the average delta CT data. So it doesn't matter what order you do this in, whether you do this first or after these two. I just wanted this to be a little bit more intuitive. So let's take a look at that. And now we can see the average delta CT has been calculated, and we also have the standard deviation for each one. So all that being said, we can now plot this. We can do ggplot, which is giving us our typical bar chart, and we can see this here. So this is now our delta CT for each of our treatment conditions, and for each of our three primers of interest. But we can also add this geome error bar using the standard deviation, and this is just how you would be able to do that. So let me add that in. Basically, we're just adding error bars now. And then finally, we can improve the look and feel a little bit by scaling the color using the Viridis package. So we run this, and then we can add our scale color viridis, and it's just a little bit nicer looking now. Maybe it's a little bit more colorblind friendly as well. So I was going to go through how to now calculate the delta-delta CT, but it's actually the same steps, just kind of in a different order. I think that the right way to do this, because we're kind of running out of time, is just to go down here to hands-on activity number 2 and take 10 minutes, just do the delta CT part for right now, get down to this part here. So that's going to be calculate delta CT all the way through till right before calculate delta delta CT. That part you can try on your own if you'd like. So let's go ahead and take 10 minutes, and then we'll come back and answer any final questions and talk about the homework for next time. Okay, so really quickly, I'm gonna just go through this and show you my answers. So the first was just kind of a recap of what we did before, where we made this tidy PCR data. You can see that here. Then I said calculate the delta CT. So the first step is to get the average CT for the housekeeping gene. So we wanted to filter so that the primer was the housekeeping gene. And then we needed to group it by, what am I thinking of here? I guess this would be by treatment. And then this would be the summarization to give you the, you could just say HK gene if you wanted here. So I've got my control RNAi1 and RNAi2 housekeeping gene values. I think that's right. Let's take a look at tidy PCR and double check. Ah, my mistake. We actually wanted to group by sample because we can see we've got our replicates here. Sorry about that. All right. So now we've got our housekeeping gene by sample. Then we wanted to calculate delta CT. So we're going to filter again for the samples that are not the housekeeping, that are the primers that are not the housekeeping gene. We're going to do a left join or a full join or a right join. They all would have worked here actually. Using housekeeping gene and tidy PCR. And then we're going to do our mutate. Right. Because I called it something else up here. So I could either make this HK gene or I could make this HKG, and that would have worked because it's the same column in both places. So we can now see we've got our delta CT, and we can get the average expression, the average delta CT, and the summary statistics using the standard deviation. So to actually get the average of the delta CT by sample or by treatment, in this case, we want to use treatment, it looks like. So what we're going to do is we're going to do a group by. And then we're going to do a summarize. So let's take a look here. See, now I've got my average delta CT by treatment. But in this case, it probably makes more sense to do sample. You could do it either way, but I think in this case, sample probably makes a little bit more sense. And then we want to actually do, again, our group by, and we want to, instead of getting the mean, we want to do the standard deviation. Mutate, getting the standard deviation. And we also want to do our join so that we can join our mean with our standard deviation now. So we can have, because we're adding our average delta CT from up here to our original data and then calculating our standard deviation. Finally, we're plotting it. So this is our ggplot. X, we can make X the sample. Y can be the average delta CT, from up here. And then we can do our geome call. So if we just take a look at these first two lines, we can see here our My mistake. It appears that I didn't include that data as I should have. So we've got our treatment here. We've got our treatment. I see what happened. So delta CT by sample, average. By sample. Then get the average. So I don't know what I did wrong here. But since we're running out of time, I will have to just put it in the video later. So go back, look at the YouTube video, and that will actually be there. I suspect that what happened is that I don't know what I did wrong. Sorry about that. All right. So let's go ahead and take a look at this now. Oh, gosh. Well, for the sake of time, I'm not going to, unless someone in the chat has the answer to this, because I don't want to try to debug this in front of you. Unless anyone wants to watch me debug this. So let me give you the homework for next week, and then I'm going to show you how to debug this for anyone who wants to stay late. Because honestly, I think that's a pretty good skill to have, and I think it's worthwhile to show you how I do it. Simon says line 288 treatments not capitalized. Yeah, that could be the issue, but I don't think it is. Let's talk about the homework and then we'll go through this. So the homework is to get started, because this is now officially the end of our R programming and basic R data science tutorial. We're now going to get started with We're now going to get started with the RNA sequencing portion of the workshop. So this is where you actually get to apply these skills to analyze some RNA-seq data. And so next time, Dr. Chen is going to be lecturing on how to analyze RNA-seq data using DE-seq2. And then we're going to be going forward with some plotting and visualizations with Dr. Bunnick's lecture, and then how to make sense of your data using methods like gene set enrichment analysis. And I'm going to be helping to teach that part with Paul. So now that we've reached the end, the homework is to get started with the RNA-seq using R in Bioconductor course in DataCamp. So this is assigned to everyone. You'll find it in your assignments. And if you can get that done before next week, that would be fantastic, because that will give you a really solid foundation for what we're going to be doing with Dr. Chen's lecture and activity. So, okay. Let's go ahead now. So for anyone who doesn't want to stay for the debugging, you can go ahead and we'll see you next time. For anyone who wants to stay for the debugging, I'm going to show you how to do that now. So how do you actually think about debugging? Well, what's the error? Well, let's go down our list of commands here. So we made our data. We calculated our average CT. And we can see this here. We go through every step. We verify that our results are what we expected. So we calculated our average delta CT, our average CT for all the housekeeping genes for each of our samples. So we can see this here. So let's go to the next step. The next step was to calculate delta CT. So let's see what happens when we do this. So we can now see that we've got our samples and we've got primer and treatment information here. So if we look here, we can see that our samples are matched up with the treatment, and that's what we expect. And we can see here that our delta CT is what we would have expected, or sorry, our CT is what we would have expected. But, and our delta CT is also what we would have expected, because we can see here for each of these that we have the correct housekeeping gene CT and the correct, yeah, so I'm actually not sure what the problem was then. So let's come down to the next part then. So delta CT being piped to group by, so we're grouping it by sample. So let's look here, that will group it by sample. And we can easily see these samples here. So we're grouping it by these samples. And then we're going to summarize. We're going to get the average delta CT. So that's going to be the mean of the delta CT column. So that's going to be the mean of, for example, it's going to be this plus this divided by 3, this plus this divided by 3, and so on and so forth. So let's ask a question here, because this doesn't seem quite right, does it? We asked it to do a summarization on delta CT. Now, delta CT had a lot of other columns, but average delta CT only seems to have sample and AVG DCT, which is the new column that we created here. So, bearing that in mind, can anyone tell me what the problem might be with my right join? And if anyone has an idea, you can unmute. Yeah. Simon, can you, if you don't mind unmuting, could you actually explain that a little bit?
Yeah, so for some reason, a bunch of your columns went to the, to the ether. And so it tries to join on a column so it'll match the whatever treatment column or something. It'll match your 2 data frames on that column. But if that column doesn't exist in one of your 2 data frames, it's got nothing to match to, and it'll throw an error.
Right, exactly. So there's a couple things that happened here. The first one, the most immediate problem, is exactly what Simon just said. So we tried to do a right join. And actually, I'll isolate this so you can see precisely what the problem is. So our right join, we've got our X is going to be our DCT data frame. Our Y is our AVG DCT data frame, or Tibble, depending on how you think about it. Now we're going to attempt to merge these 2 data sets based on these columns. So what does this imply? Well, it implies that we believe all 3 of these columns belong in both data sets. Both data sets contain all 3 of these columns. Is that really true? Well, let's look at DCT. DCT contains sample, primer, and treatment. Let's take a look at AVG DCT. It only contains sample, and it doesn't contain any of the other ones. So it doesn't have primer and it doesn't have treatment. So basically what Simon said here, it wants to join on a column that no longer exists. That's because all that exists now in both data sets is the sample column. And now this works because both data sets had this sample column. So what happened when we tried to join on all 3? It said join columns must be present in data. Problem with primer and treatment. So that was basically its way of telling us that we messed up by trying to give it columns that weren't in both of our data sets. Does anybody have any questions on why that doesn't work and why this gave us an error? Okay. Well, yeah, so that was going to be my next point, which is why did my columns disappear in the first place? Just out of curiosity, does anybody have a guess as to why? So G1 said because you had group by with only sample. So I don't think that's the answer. I'll admit, like, I'm like only about 90% sure that's not the answer. But yeah, maybe 10% you could be right. But no, I don't think that's the reason. Because we've had other situations where you've been able to group by without losing other columns. Does anybody have any other ideas? So let's try, because like when you're debugging something, at the end of the day, you don't really know the answer yet, right? So let's try Jiwan's suggestion here. So let's try grouping it also by treatment. So we were able to keep treatment this time. So yeah, I guess that does work then. If you do keep, oh wait, hold on, what am I thinking? No, that won't work because now you're grouping it by. Well, I guess technically that does work because treatment is a redundant group, right? Because all of your control ones are also in the control treatment group. So yeah, this technically does work. It wouldn't be my first approach, but yeah, it does technically work. But let's try something else. And I'm trying to think of like what the right way to do this is. So let's come back all the way up to here when we first made our DCT. Let me rename this. I suspect this could alleviate our problem. Oh, never mind, it doesn't. Oh, right. Let's see here. Now we've got DCT and let's see here. Yeah, so I guess actually I think G1 was right. I think that's pretty much all you had to do then was make sure that you include your other columns that you want to keep. So if I group by sample, or if I keep sample, if I keep treatment, and if I keep DCT call, let's see what happens. So it still got rid of them. But if I put these in the group by, now let's see. So it did keep it, just like we thought it would. And primer. Yeah, so Simon has a good point. And actually, that's now my confusion. So again, this isn't really a lecture anymore. This is just me showing you how I would think about stuff like this. So I'm going to basically show you how my thought process works when I run into a confusing situation like this. So in my mind, grouping by something and summarizing shouldn't get rid of your other columns, unless there's some good reason why it would collapse them. So let's actually now check our cheat sheet. So I put this in box. This is the, whoops. This is the cheat sheet for data wrangling in R. So basically, when you look at the example here, so this is basically showing you an example of a summarized statement using the iris data frame. So we can see here that it only returns for us the one column that we made, but it actually ditched all the other columns. So I suppose that does mean then that, yeah, I mean, I suppose that means then that it does ditch the columns. And then just to show you, like, again, my thought process on this, my next step will actually be to like Google dplyr group by summarize removes columns. And there's good old Stack Overflow. And this question has a lot of, I also have this question, so I upvote it. So the question that they're asking here is that they're trying to group the data set based on one of the columns and take the min value of the other columns, but it's only giving them 2 columns back. So that's basically the exact problem that we're running into. So let's look at the answer. So apparently there's a couple ways we could handle this. We could use filter, we could use slice, or we could do a group by. You could also do group by without summarize. So this isn't exactly the answer that I think I'm looking for. So let's try this one. Because I don't want minimum, I want something that's going to do, for example, like a mean calculation. Yeah. Basically, what this is telling me is a few things. The first thing that it's telling me is that when you aggregate your data, you only are going to keep the columns that you did the group by with. But the other thing it's telling me is that there are ways to override that, but I'll probably never be in a situation where I am worried too much about that I'm willing to actually seek out a special way to do it. So as kind of unsatisfying as I feel like G1, like no, I'm not like, this isn't meant to be like offensive. As like unsatisfying as I feel like G1's answer is, it's probably the one that I would genuinely use. Because in one step, it makes it really simple to specify which of the columns I want to keep. And, you know, yeah, like you said, you can even put primer in here. Okay. And now that I've got primer and treatment, I can do my join like before, except that I changed this. So let me. Okay. Right. So the problem was that when you did this group by, you now had an issue where, oh, yeah. No, I know what the problem is. The problem was that I renamed it up here. So is everyone able to follow how I'm debugging this? Does anybody have any questions about what I'm doing here to get to the point where I have like a correct answer? So I've got my treatment now in my data. So this was the answer I was originally looking for, essentially. All right. So I guess I'll just ask one more time. Does anybody have any questions about how I debugged that, or how I knew to Google those things that I knew to Google, or how I made my decisions on this to get to the point where I have like now the correct answer? Or just more generally, any other questions? Okay. Well, I'm going to go ahead and wrap it up then. So if you guys get a chance, send me your practice problems from weeks 2 if you haven't already, and week 4 if you haven't already. And for those of you who have, I'm sorry I haven't had a chance to get them back to you yet. I had just not a whole lot of warning about the fact that I would be running this particular lecture, so I just have not had a chance yet to look through those, but I'll do that after this. Okay, so yeah, with all that, feel free to drop by office hours if you have any questions, and I'll see you all next time.